Morning to us students. Today we're going to continue Activity 3 from Chapter 4, Biological Identity and Genotype. To continue Activity 3, entitled The Genotype, please open your books to page 80. We're going to start with a recap of what we took from the previous video. What is a genotype? A genotype is the list of the alleles that each individual has for a specific gene. The genotype can either be homozygous or heterozygous. Homozygous is when we have both alleles identical. Heterozygous is when you have two different alleles. Example of a gene is the ABO gene. The gene codes for the blood group. Alleles are A, B, and O alleles for the ABO system. Each allele codes for a specific antigen that is expressed on the red blood cells, and the expression of this antigen indicates the blood group of the individual, except in the case of O, which causes a lack of the expression of this antigen on the red blood cells. Allele A is dominant over allele O. Allele B is also dominant over allele O. However, alleles A and B are equally dominant. That's why we call them codominant. Here is a summary of all the blood groups that human beings can have. In this table, you can see the genotype written in both possibilities, the corresponding phenotype, the justification for this phenotype, and the antigen or the molecule that is expressed on the membrane of the red blood cell. I'm going to give one example. For example, individuals having genotype AA will have a phenotype A that is expressed with the letter between two brackets. Why is it considered phenotype A? Because any of the A alleles can dominate over the other, and therefore the only antigen expressed on top of the membrane of the red blood cells is the A antigen. Another example of a gene that is studied is the rhesus gene, which codes for either a positive allele or a negative blood group allele. Whenever rhesus antigen is present on the membrane of the red blood cells, then this indicates that the blood group is positive. As shown here, red blood cells express the Rh antigen, which leads to the fact that these red blood cells are going to be considered Rh positive. Whenever rhesus antigen is absent from the membrane of the red blood cells, just like in the case of the O blood group, then this indicates that the blood group is negative. As you can see here, the red blood cells don't express an Rh antigen, and therefore they are considered blood group negative. An application for both the ABO system and the Rh system. Given the following legend, if the triangular shape codes for the A antigen and the semicircular shape codes for the B antigen and the square shape codes for the Rh antigen, solve the following three examples. In example 1, the question is, specify the blood group of this red blood cell. The answer is, this red blood cell belongs to blood group O positive, since it expressed on its membrane only one antigen, which is Rh. Neither A nor B antigens were present. In example 2, the question is, identify the blood group of this red blood cell. In identify, you're supposed to use a property. The answer is, knowing that the antigens specify the blood group. And since this red blood cell has both antigens A and Rh, then it belongs to blood group A positive. In example 3, the question is, identify the blood group of this red blood cell. The answer is, knowing that the antigens specify the blood group, and since this red blood cell has only one antigen, B, on its membrane and lacks the antigen Rh, then it belongs to blood group B-. The genotype can also be revealed by DNA fingerprinting. What is DNA fingerprinting? The DNA fingerprinting is a technique based on using DNA molecules after cutting them and letting them migrate on a specific gel where they stop at specific levels, forming bands, as shown in the next document to the right. The shown bands can either represent the entire genome, which means all the genetic material, or a specific gene. A child must share DNA bands with the mother and the father at the same time, otherwise the child is not theirs. The objectives of using this technique are to determine the paternity of a child, to determine the origin of the genetic trait, to detect genetic abnormalities, to find a criminal, and to determine the identity of dead bodies. Why do we use the term fingerprint? We use the term fingerprint because every individual has his own specific DNA fingerprint. And that's why every fingerprint is unique to every individual, except for identical twins that have exactly the same DNA fingerprint as they have exactly the same DNA. In document 1, you can see a DNA fingerprinting done for suspects 1 and suspect 2, and a sample from the crime scene was taken, and document 1 shows also its DNA fingerprinting. By comparing the DNA bands of the two suspects with the DNA bands of those obtained from the crime scene, the suspect 2 is most probably the criminal, as all his bands are common to those found in the crime scene, and therefore he is the individual that was present at the crime scene. Kindly to us students, copy the slides and the content of this video clearly on your copybooks and study the content for next time, and we'll continue this activity in the following video next time.